I hope you've been enjoying the first day of MDF's first virtual conference. I'm Allison Formal, the Senior Science Advisor at MDF. Welcome to this special time in our annual conference when our industry partners come together to share updates about their myotonic dystrophy programs and their cutting-edge work in the development of different therapeutics. This is a chance for us to hear directly from our partners about their approach, their process, and their progress. Today we will hear from Avidity Biosciences, Ionis Pharmaceuticals, Dyne Therapeutics, and NeuBase. Unfortunately, we're short on time, so we will not have the opportunity to ask speakers questions during the session. However, many of our industry partners have exhibit booths, which will be open for live chats until 6 p.m. this evening. So if you have follow-up questions after this session, go to the Exhibitors tab on the menu bar of the conference website. Select Conference Exhibitors and click on the company that you're interested in connecting with. Once you are on the company's page, there will be a button that says Join Live Showcase Now. Click that button to join the conversation with the company. Thank you so much for attending the conference. We hope you have a wonderful rest of the meeting. Thank you.
I'd like to thank the organizers for inviting me here today. My name is Art Levin, and I'm the Chief Scientific Officer at Avidity Biosciences. I'd like to talk to you today about antibody oligonucleotide conjugates for the treatment of myotonic dystrophy type 1. Avidity Biosciences is a biotechnology company located in La Jolla, California. We are using a novel technology called antibody oligonucleotide conjugates. It's a novel class of RNA targeting therapeutics. And I know that's a mouthful, but we'll get to that in a second. I may be making some forward-looking statements during the course of this talk. What is an antibody oligonucleotide conjugate? Let's break it up into its component pieces. Starting from the left-hand side of the slide, you'll see that small red object. Looks like a pig's tail. That is a double helix drug called an siRNA, a short interfering RNA. Oligonucleotide simply means a few nucleotides. Generally, in RNA DNA, there may be thousands or even millions of nucleotides joined together to make up the genetic information. These particular drugs are actually small bits of nucleic acid, about 20 nucleotides long, and that's why they're called oligonucleotides. The oligonucleotide is designed specifically to bind to a site on the disease-related RNA that causes myotonic dystrophy type 1. It binds with Watson and Crick base pairing rules. If that brings you back to high school, I'm sorry, but we are involved here in a very interesting scientific process where we've designed molecules to specifically bind to a region of the disease-related RNA, and everywhere there's an A, a T will bind, and everywhere there's a G, a C will bind. And so by making a very specific code of RNA, you're going to only bind to the disease-related protein or disease-related messenger RNA, and when that happens, it's going to call in an enzyme which will then destroy that messenger RNA. So that's the oligonucleotide part. The antibody part is interesting because we're using that antibody as a Trojan horse. Antibodies, as you know, are proteins that the immune system can make in order to target specific disease-related proteins. In this case, over the past 30 years, we've learned how to make monoclonal antibodies to almost every protein. So we've engineered a specific monoclonal antibody that's going to bind to an important protein on the outside of the muscle cell. And the muscle cell is going to think that it's taking that specific protein and the antibody that's bound to it up, thinking that it's taking up iron to feed itself, when in reality it's going to be taking up our small piece of oligonucleotide, our little oligonucleotide therapeutic. And in the course of that, we're going to get the therapeutic activity. So what you can see on this diagram is we put those components together. We put an oligonucleotide along with a molecule that has been exquisitely designed in order to bind specifically to a protein called the transferrin receptor that exists on the outside of every muscle cell. And when the muscle cell thinks it's pulling in that transferrin receptor, taking in iron, it's actually going to be taking in our drug, hence the Trojan horse. So the red bit is the warrior, the blue bit is the horse. And in between there, you can see a small yellow tail, and that's the linker. The scientists at Avidity Biosciences have engineered each one of these components specifically to perform the functions that we need in order to produce the best potential therapeutic agent to target the disease-related gene in myotonic dystrophy. And so what you see schematically here on the left-hand side is in circulation is our antibody oligonucleotide conjugate. It's looking for that little protein that exists on the outside of that muscle cell. And the antibody oligonucleotide conjugate, the antibody itself, is a remarkable molecule because if there are 1 billion protein molecules in solution, it will find the right one every time. It has an extraordinarily high affinity and a high selectivity for the right cell surface protein. Once it binds to that cell surface protein, as you can see on the right side of the slide, the AOC is then taken up. Again, the cell thinks that it's taking up iron, and in fact it engulfs the protein, and ultimately the oligonucleotide is released from the antibody as the antibody is destroyed in that particular vesicle that you see there, and it releases the oligonucleotide therapeutic to allow it to perform its function. And on the next slide, what you can see here is that we're looking to target a specific gene product called the messenger RNA for a gene called DMPK. That particular RNA in the disease state has an expansion of a triple nucleotide repeat. So there are too many repeats, these happen to be CUG repeats, that are in this messenger RNA. People with myotonic dystrophy can have thousands of copies of these CUG repeats. People without myotonic dystrophy may only have 10 or 15 copies. Those thousands of copies of CUG repeats actually induce a toxicity. And the way that they induce a toxicity is those CUG repeats will bind to one another, and then they will attract a very important protein for messenger RNA processing called muscle blind-like protein, here abbreviated MBNL. When the MBNL binds to the CUG repeats, the excess number of CUG repeats that a patient with myotonic dystrophy has, the MBNL cannot perform its downstream function. And the downstream function of MBNL, or the actual work that MBNL does in the body, is to process other proteins. And these other proteins are important in a number of different disease processes, including how your heart rhythm works, or how your muscles contract. And so if these proteins are not being processed properly, you have the changes that are associated with the disease. What our drug does is it comes in and it binds to an enzyme complex called RISC, the RNA inducible silencing complex, and that's shown schematically as that funny group of shapes on the left-hand side. It will bind to that particular enzyme complex, and that enzyme complex goes looking for the perfect match for our particular RNA therapeutic on the messenger RNA that's associated with the disease. And when it finds that perfect match, the enzymes associated with that complex will destroy the messenger RNA, releasing the MBNL. So in the schematic, it goes from a red MBNL that's not functioning to a green MBNL that can then go downstream, look for the genes the messenger RNAs that it's supposed to be processing, and it will ultimately change the course of the disease because the proteins which were previously misplaced over time are going to be replaced with proteins that are properly processed. And that's really the key to the therapeutic that we're dealing with here. So here you can see that a single dose of an antibody oligonucleotide conjugate that was given intravenously to non-human primates, which is a fancy word for monkeys, after a single dose of 2 milligrams per kilogram of the oligonucleotide, you can see that we get a 75% reduction in the levels of the DMPK RNA. And if you reduce the DMPK RNA, you're going to be reducing the number of CUGs that are in the nucleus. You're going to be freeing up muscle blind-like proteins. The really remarkable thing about this particular figure is, look at the time course. As you move out, the first data point we select is actually at 4 weeks after a single intravenous dose. The next data point is at 8 weeks after a single intravenous dose. And finally, the third time point is 12 weeks after a single intravenous dose. And you can see that a single dose of this drug has tremendously long and durable activity in the non-human primate, which speaks really well to the potential for this drug to be utilized as a relatively infrequently given drug, perhaps as infrequently as quarterly or once every 3 months or even less frequently. Of course, we talked about the cardiac effects and the effects on breathing. So the question is, if you can reduce the expression in skeletal muscles like the quadriceps and the gastrocnemius, the quadriceps and the calf muscles of these monkeys, can you change the expression of DMPK in other muscles like the heart or other muscles like the diaphragm? And what you're seeing on the left-hand side of this slide, in fact, is a study that we did again in non-human primates or monkeys, where we gave single doses of either 1, 2.5, or 5 milligrams per kilogram. And you can see that even at a dose of 1, there's a 50% reduction in the expression of the DMPK. So 50% of the CUGs that would be associated with that DMPK are now destroyed and are not present. On the right-hand side, again, just to emphasize the point, you can see that at 12 weeks in the heart and diaphragm, there's still greater than 50% reduction in the expression of that target gene. Again, suggesting that when we get to the clinic, which will be soon, we will be able to dose these drugs relatively infrequently, as I said, quarterly or even less. We've spent the past year working diligently to increase our knowledge of the antibody oligonucleotide conjugates for this purpose. We've been able to demonstrate a favorable preclinical program. We are going to use, as I said, relatively infrequent dosing, and we're getting ready to move into a clinical trial at a phase 1, phase 2 clinical trial in 2021. We're also partnering with the MDF to get you as much information as soon as possible. And to that end, we have a booth, a virtual booth at the meeting where you can ask questions, because I'm sure there are a lot of questions. I went quickly through a lot of pretty technical science here. I'll be happy to address those personally, and it would be my pleasure. There's a big team of people that have worked with me on this particular project, and they're shown here, and I'd like to thank you for your attention.
Before I begin, I want to thank the Myotonic for creating this wonderful virtual opportunity for us to connect with all of you. I'd like to welcome all the families that are joining us here today. It's a great chance for us to spend a few minutes and introduce you to Ionis, who we are, our vision, and our commitment to science and medicine, and most importantly, you, the patients that we are here to serve. My name is Kristina Bowyer, and I'm the Vice President of Patient-Centered Drug Development. And my group is really focused on partnering with the patient community in order to make sure that we incorporate your voice and your perspective into every aspect of drug development. Ionis is a small biotech company. We were founded in 1989, and we're located in Carlsbad, California. We've pioneered a technology that we believe would change the future of medicine, and we have stayed small and laser focused over the years in order to be the best at what we do, so that we can make the best drugs we can to help patients in need. We have quite a large pipeline for such a small company and have multiple drugs in clinical trials, targeting a wide range of diseases, including quite a number of programs that are looking at neuromuscular disorders. We're applying pioneering advances in antisense to neurological disease drug development. We're focused on the chemistry to support the creation of better drugs, and we're applying our advances to the basic research to identify drugs which can lead to increased patient convenience. And we're incorporating what we learn from patients into our development programs so that we can efficiently continue to create life-changing therapies that are focused on alleviating patient needs and concerns. Our success is now evident in the multiple approved drugs that we have for severe and rare diseases, such as Spinraza, which was the first approved drug for the treatment of spinal muscular atrophy. Tegsedi, a transformative medicine for patients with hereditary polyneuropathy, a type of amyloidosis. And Waylivra is approved in Europe for the treatment of patients with familial chylomicronemia syndrome. These are patients that are plagued with life-threatening triglyceride levels. Our antisense drugs for myotonic dystrophy type 1 are designed to reduce toxic mRNA levels in all tissues. Therefore, they have disease-modifying mechanism of action targeting DMPK in the muscle and central nervous system. Those are the two areas that we are focusing our research. The mechanism of action will be the same no matter what the target tissue, because RNase H1 mediated degradation of DMPK basically breaks apart the RNA and it releases sequestered proteins from within the toxic RNA, as I'm sure you heard about in everybody's messages on the role of toxic DMPK accumulation, and with that release of sequestered proteins, it restores a cell so that it can go back to its normal cellular function and splicing patterns. We are committed to myotonic dystrophy, and we're focused on the chemistry to support the creation of new drugs, which is why we are targeting both a muscle and a CNS drug moving forward. We're leveraging 30 years of knowledge and experience, and with that, we're screening thousands of oligos or ASOs right now to find a compound to treat both aspects of myotonic dystrophy type 1. We're incorporating what we learned from patients to develop our program so that we can efficiently continue to create life-changing therapies for you. So where are we today? We are pursuing the identification of lead drugs that target both the muscle and CNS, DMPK. We have an extraordinary partnership with Biogen focused on severe and neurological disorders, and together we are dedicated to advancing the myotonic dystrophy program. Our goal is to create the best drug we can that serves the broadest DM1 patient population. We're committing to making a difference for the future of patients with myotonic dystrophy. Ionis is committed to the science, to medicine, to patients. We are here to create transformational medicines for patients in need, and we want to work with you to incorporate your voice and your goals for a treatment into our development program. You're our main source of inspiration. And with that, I want to thank you for inspiring us every day with your strength and persevering. We are here to be a force of life for you, and we look forward to seeing you in person next year at the Myotonic Conference. Thank you so much for joining us. And for more information, please feel free to visit our virtual booth or email us at patients@ionisph.com. Thank you.
Hello, and thank you for joining us today. We are excited to share Dyne's research and drug development advances as we finalize our drug development and clinical trial program for myotonic dystrophy. I'm Romesh Subramanian, Chief Scientific Officer and founder of Dyne. Please don't hesitate to visit our virtual booth after the session if you have questions or want more information. Just a reminder that we will be making forward-looking statements in our presentation that involve certain risks and uncertainties. Dyne is on an inspiring mission to deliver life-transforming therapies for people with serious muscle diseases. Oligos are a proven therapeutic approach for liver and brain-related diseases, tissues where significant amounts of medicine have successfully been delivered. Despite clear understanding of how to treat myotonic dystrophy and well understood disease biology, we still do not have effective therapies for myotonic dystrophy. This has largely been an issue of delivery and inability to get the therapy to the affected muscles. So we are innovating delivery methods to bring modern oligos to muscle. I'll start with an overview of our DM1 program. This is Joachim Beckelman, a DM1 community member and friend of the Dyne family who was diagnosed with DM1 in his 30s. As you know, the genetic basis of the disease is expansion of a CTG triplet repeat that forms toxic RNA that gets stuck in the nucleus of muscle cells. This shuts down critical channels and muscle cell functions, resulting in disease symptoms such as myotonia, muscle weakness, cardiac, pulmonary, and GI issues. The need for effective therapies is severe, as there's a large population of people living with myotonic dystrophy around the world. We truly believe the Dyne's Force platform is the opportunity to stop and also reverse disease progression. The foundation of Dyne's approach is the Force platform. Dyne's Force platform makes it possible to deliver modern oligotherapies to muscle diseases. Force consists of three parts: an antibody, linker, and the payload, which is the actual therapy or drug. Our proprietary antibody is a Fab or fragment antibody, which binds to the transferrin receptor or TFR1. This receptor, which is on the outside of muscle cells, serves as a doorway into muscles. The Fab antibody is our key to unlock the door and effectively deliver nucleic acids to different muscles in the body, such as skeletal, cardiac, and smooth muscle. We use a clinically validated linker to connect the Fab to the payload or drug. We selected the Val-Cit linker as the linker for our Force platform, based on its clinically validated safety and efficacy in approved products, as well as its serum stability and its endosomal release attributes. Our payload needs to decrease toxic DMPK RNA to modify disease, and hence we use an ASO which targets the toxic nuclear RNA. I'll briefly describe DM1 disease pathophysiology and then present our data validating Dyne's Force platform. DM1 is an autosomal dominant triplet repeat disease of the DMPK gene. These repeats, when transcribed, form toxic clumps of RNA in the nucleus. In DM1, the higher than normal number of CTG repeats form large hairpin loops that remain trapped in the nucleus, forming nuclear foci that bind splicing proteins and inhibit their ability to perform normal cell functions. This leads to the clinical symptoms for DM1, which are muscle weakness, myotonia, cardiac arrhythmia, or pulmonary abnormalities, which are the leading cause of death. The Force platform targets this disease upstream in the process at the toxic RNA which is stuck in the nucleus, and thereby we address the genetic basis of DM1. We rationally chose the single-stranded ASO to target nuclear toxic RNA. We believe this approach has the potential to stop DM1 progression and reverse the disease. So let's take a look at the data utilizing Dyne's Force platform. When toxic nuclear DMPK levels are reduced, the nuclear foci are diminished, releasing splicing proteins, allowing restoration of normal RNA function. As illustrated here, in this study in affected DM1 patient cells, the left panel shows multiple red foci or dots within the blue nucleus following treatment with saline or PBS, which is the control treatment. A single dose of Dyne's Force platform reduces nuclear DMPK foci, as demonstrated by the reduction in red foci in the right panel. In a DM1 patient cell line with over 2,000 repeats, we observed release of important MBNL splicing proteins from the hairpins and correction of important cell functions with a single dose of Force. You can see from these charts that once we get our therapy into the cell's nucleus, it begins to correct the splicing problems. In the black bar is the untreated cells, and we observe about 10% splicing. A single dose of Dyne's molecule, seen in blue, increases splicing to 30%, which is a significant improvement. A single dose of force thereby targets toxic DMPK RNA in the nucleus and corrects splicing. We advanced our molecules into a well-validated DM1 animal model called the HSA-LR mouse. HSA-LR mouse model has long hairpin repeats in the nucleus. It creates the protein missplicing that you observe in affected people and is one of the very few animal models that includes a symptom experienced by people with DM, which is myotonia. As shown on the left panel, we observed correction of splicing in multiple muscles, including leg muscles such as the gastroc, TA, and quadriceps muscle, and thereby reversed myotonia as seen on the right panel. For those of you interested in the scientific aspect, the left-hand side y-axis shows the missplicing that happens in affected cells, where 1 is high amount of missplicing, and 0, where green line is unaffected muscle splicing. The red circle is vehicle-treated cohort of mice. Green represents 10 milligrams per kilogram of Dyne molecule, and blue represents 20 milligrams per kilogram of Dyne molecule administered IV. We observe a correction towards unaffected splicing with a single low dose of our molecule, which improves as the dose is increased. Each of these points is a composite of 35 RNAs and the splice score for each of them, demonstrating that we are normalizing multiple muscle proteins in an animal. The profound splice correction with a single low dose on the left panel results in the significant decrease in myotonia observed in the right panel in the very same mouse model. These studies were conducted blinded at a third party, and they demonstrate that a single dose of force nearly reversed myotonia in 3 separate muscle groups. On the right, as you can see, the untreated black bars did not alter myotonia. That's a control treatment. Red is naked ASO, which decreased myotonia slightly, and blue is our Dyne molecule, demonstrating reversal of myotonia in vivo. Previously published data from another company demonstrated changes in myotonia after 1 month of dosing. However, it needed 8 doses, and each dose was 25 milligrams per kilogram. This demonstrates the value of our force platform, which is targeted delivery of low doses of oligo, infrequently into the muscle, and correcting splicing and disease symptoms. In summary, we've shared data demonstrating that our force platform targeted toxic DMPK RNA in the nucleus, reduced nuclear foci, corrected splicing changes, and reversed myotonia after a single dose. Last year, we also shared with you that our compounds deliver DMPK targeting ASO to mouse and monkey tissues. We also observed durable DMPK RNA reductions up to 12 weeks, and our therapy is well tolerated. Most importantly, we have the right team to execute on our strategy and deliver to fulfill our mission of transforming the lives of patients with DM and other serious muscle diseases. This is a picture of the team with Joachim, who we mentioned previously at our Rare Disease Day event earlier this year. We have a shared commitment to realize the full potential of our FORCE platform and deliver for DM patients. With that, I thank you for the opportunity to share Dyne's compelling data with you today. Again, please visit our virtual booth after this session to ask questions and learn more. We look forward to talking with you. Thank you.
My name is Dietrich Stephan. I'm the CEO and chairman of Newbase Therapeutics. Newbase is a pharmaceutical company that is working to develop a new type of synthetic medicine to address myotonic dystrophy type 1. We are at a unique point in history that promises great hope in the near term for individuals suffering from myotonic dystrophy. Historically, the way therapies were developed was to take many, many, many small chemical molecules and screen them against cells from patients with disease in a trial and error manner. This was a very expensive and protracted type of therapy development endeavor. I often refer to it as a bespoke, handcrafted era of therapy development. Today, as a result of the sequencing of the 3 billion letter human genome and literally trillions of dollars of investment in computational technologies, we can develop drugs that specifically engage with a misbehaving gene and silence it before it manifests itself in a disease. And that is precisely the strategy that we are using at NeuBase Therapeutics. The type of therapy that we are using is developed on a synthetic scaffold, so a short polymer. And into that, we can program the precise sequence of A, C's, G's, and T's that will match or complement the target gene of interest that we want to silence. And because we use a single scaffold that's inert, precise, and easy to deliver, we have the potential to not only move forward expeditiously in addressing myotonic dystrophy type 1, but also provide a scalable therapy development platform for other diseases. Remember, every human disease is genetic, and thus, if we perfect the scaffold, we have a scalable potential that uses nature's digital information encoding scheme, the human genome, letters of A, C's, G's, and T's in different combinations that stretch for 6 billion base pairs, 3 billion from mom, 3 billion from dad. And now we can use that and reverse engineer it to create better therapies. We've put an incredible team around this to make sure that we develop the technology to its fullest extent. And just to give you a flavor of that team, there are individuals who were involved in sequencing the first human genome almost 20 years ago on our team. There are individuals who pioneered personal genomics and interpretation, so individuals could manage their own health portfolios moving forward. A member of our team co-invented human genome editing. A member of our team ran the $5 billion a year National Cancer Institute, which is the largest funding organization for cancer research in the US. We have members of the National Academy of Sciences, the National Academy of Medicine, and the National Academy of Engineering. And members of our team were instrumental in turning HIV infection from a lethal disease to a chronic disease. So we have a good team, and we're aiming all of this firepower at myotonic dystrophy. So how can we at NeuBase help individuals who are suffering from myotonic dystrophy type 1? First, it's important to take a step back and remember what is driving this disease. One copy of a gene named DMPK contains extra genetic material, and that extra genetic material is in the form of what's called a trinucleotide repeat. The letter CUG repeated over and over and over again. And when that repeat gets too long, in the intermediate form of that gene, the RNA molecule, which is the copy of the gene at the DNA level, that repeat forms a hairpin structure. And that becomes a magnet for proteins in the cell. And when those proteins stick to the hairpin, they can't go do what they're supposed to do. In fact, that causes these RNA molecules to also aggregate in cells of people with DM1, and those can be seen under the microscope. So what our therapies do is interact with that hairpin. And when they interact with that hairpin, they kick those stuck proteins back off to go do what they're supposed to do. Importantly, and this is different from some of our colleagues developing other types of solutions, we do not get rid of the DMPK gene, nor do we get rid of the DMPK protein. And we believe that's important because nature has left that gene on for a reason. And we know it's critical for regenerating muscle and at the neuromuscular junction where motor nerves from your spine connect to muscle and allow you to move. So turning that gene off may have downstream consequences that are not great. And that's an advantage of our strategy. So again, we're using a scaffold that's synthetic, and into it we are engineering nucleobases, A's, G's, and T's that match that hairpin. And this is a visual just to help you get your head around what we're specifically doing. So at the bottom of this slide, you see a cartoon of our therapy. The squares are the synthetic backbone or scaffold, and the triangles are the A's, G's, and T's. We have shown that we can let that drug loose via a systemic administration, intravenous or subcutaneous, and it can get to the tissues that we know are where the disease manifests: skeletal muscle, heart, brain, diaphragm, etc. So the scaffold is easy to administer. It's precise in that it's more specific for the gene that's misbehaving versus the unaffected copy or other copies in the genome. And it's inert, which is important if one plans to take a therapy like this for the rest of one's life. And again, it does not, importantly, degrade the resultant protein, which some of our colleagues focus on. I want to remind you, as if you didn't know this already, DM1 is a whole body disease, and having a therapy that's accessible to all of the tissues that are affected is important. So what have we shown and where are we going? We have shown that our drugs can get to where they are needed in the bodies of patients with DM1 after a systemic administration. Secondly, we have shown that our therapies work, that they can silence the misbehaving gene and act at the causal level without interacting with other genes that are important in the body. And finally, our synthetic scaffolds are well tolerated. And that's important, again, for a lifelong solution. Over the next 12 to 18 months, we will be driving into clinical trials and quickly, hopefully, emerging into the marketplace with our solutions. If you have questions about what we're doing, please feel free to visit our website at NewBaseTherapeutics.com. We have a patient portal there as well. You can also find updates on Twitter at NewBase Inc. or on LinkedIn. And finally, please don't hesitate to reach out directly. We would love to engage with all of you. Our email address is info@newbase.com. Thank you, and please don't hesitate to stay in touch. We look forward to working with all of you as we move forward.